Good morning, everyone. Good evening, everyone. Please confirm if you all can hear me. Am I audible to everyone? Please confirm. Good evening, Aishwarya. Can you hear me? Is my voice loud and clear? Good evening, Rahul. Good evening, Anusha. Good evening, Aishwarya. Okay, thank you all for confirming. Good evening, Surendra. So we'll start the class in a minute. Let's everybody join. So how are you all? I hope everyone is doing great. Are you all revising genetics? So far we have been discussing questions from Unit 8, that is inheritance biology. And in every session of from every session of from Unit 8, I was discussing the topics, I was discussing the questions on particular topics, right? But today, in this session of Brahma series, I will be giving you mixed questions as you get in CSIR NET exam, right? So far, you know that, okay, today we will be discussing questions on probability. Today we will be discussing questions from subunit B, that is concept of gene. Then from C, that is extensions of Mendelian principle. Then from D, subunit D, linkage, tetrad analysis, right? But today I will be giving you the questions from subunit A to E, topics from subunit A to E of unit 8 only, but it will be all mixed as you get in CSIR NET exam. So this is just for your practice, like as you have to, like if the questions comes from unit 8, like maybe one from linkage, one from probability. So in that case, will you be able to, like you need to find out that whether you are able to recall the formulas, you are able to recall the concepts or not. So I hope everyone has joined the session. So let's get started. And I'm sure now you all are familiar with the concepts, the strategies of solving questions, and the formulas. But still, if you have any doubt, please let me know. And no worries, I will be giving you questions for practice. But still, if you're not able to solve them, no worries. I will be helping you in solving the question once you try yourself. So let's start with the very first question of this session. The question is a phenylketonuria. It's a metabolic disease in humans. It is caused by recessive allele K. If two heterozygous carriers of the allele marry and plan a family of 5 children, what is the chance that first child will be an unaffected girl? So go through this question. I'm sure while going through this question, you have understood that this question is based on probability. So just recall what I have told you so far about probability questions, probability based questions. Just recall the concepts, recall the formula, and try to solve the question. Yeah, very good evening, Maheshwari and Shivangi. So you all can solve the question. I'm giving you some time to solve. And then in the chat, you can let me know what is the answer. And I'll let you know whether the answer you have, the question you have answered is correct or not. And even I will be explaining you the answer of the question. Okay, Surendra, very quick. Option 3. Okay, Lakshmanan, yes, basically the students who are with us from the beginning, for them I have already completed the probability. You can go through the recordings also. This is why we always keep a recording. And no worries, like once we are done with the topic that we are doing in the class, I will again discuss the probability in the class. Okay, for you and the new joinees which are there. Don't worry. Okay, Rahul and Sajanya, you have also answered option 3. Fine. Please, everybody, try the question, try solving this question. See, this is the Biotechnica has come up with this Brahma series to help you all, right? And at the end, you need to understand that at the end in the exam, you have to solve the questions, right? So at least try. Even if if you are not able to get the correct answer, no problem in that. You are still in the learning phase, right? But first, you need to try. If you don't try, then how will you know that? Whether you are able to solve such questions or not. And believe me, once you start solving these questions and getting the correct answers, automatically it will give you confidence, right? So, and obviously that will again generate a lot of interest in solving, like it will generate an interest in solving these questions. Okay, so quickly solve. See, if you give me the answers quickly, more questions I can solve in the class, right? What's the use if I just give you the answer, explain you and give you the answer and go, right? At least you need to try first. Okay, I can see some more have joined now. So quickly give me the answer of this question. Okay, Aishwarya, you are confused in option 2. Fine, no worries. So see, as I always say, read the question carefully and try to understand the question. If you're not able to understand the question by reading it once, try to read it again. So in the question, it is mentioned what all information is given to us in the question. It is mentioned that phenylketonuria, it's a metabolic disease in humans, right? And basically, it is associated with a recessive allele. So one thing is very much clear that if any disease is associated with recessive allele, it will be expressed only when the recessive allele comes in homozygous condition, right? So this is one information which is given about the disease. Second information given is that the two heterozygous carriers of the allele marry and plan a family of 5 children. So that means here the parents are heterozygous. So if we consider, like if one is recessive allele, another will be dominant. And if recessive allele is responsible for the disease, dominant will be for the normal phenotype, that is without the disease. So let's assume the uppercase letter K as the dominant allele and lowercase letter k as the recessive allele. So parents here are heterozygous. So they are having this genotype, both the parents. And in the question, it is asked that what is the chance that first child will be an unaffected girl? So that means you have to find out the probability for getting an unaffected child, and also you need to find out the probability of getting a child which is specifically a girl child. So here you need to calculate the probability separately for unaffected and separately for the girl child. Now, this is what is given in the question. Now, what you need to recall here. So once you understand the question, you need to find out the probability. And second thing is you have to find out which rule you have to apply here. So here, this here as you can see, these are the parents, right? So parents are heterozygous. Now, parents are heterozygous means that they will not show the disease symptoms. They will be the carrier because they will be carrier of the diseased allele, but they will not themselves show the symptoms. So basically, the family, so these parents, they plan to have a 5 children. So if I am performing this kind of cross, definitely, I'm sure you know how to determine the genotype in the progeny, right? So you know either we'll get homozygous, the child will have homozygous dominant condition or heterozygous condition, or the child will be homozygous recessive. And in homozygous recessive only the phenylketonuria, that is the disease, will show, right? So here, if I have to ask you what is the probability of unaffected? So you know if the child is having homozygous dominant condition or heterozygous, the child will be normal. That is unaffected. So child will be unaffected, probability is 3 by 4, and probability of getting a child which is affected is always 1 by 4 when we are performing a cross. So this is like a monohybrid cross. And So this is one thing we have calculated, that is probability of affected child. But what is the probability of getting a girl child? So you know that the female can contribute only X chromosome, but a male can contribute X or Y. So if both the parents are contributing X chromosome, the child will be a female. But if a mother is contributing X and father is contributing Y, then it's a male child. So probability of getting a male or a girl child is 50-50. That is half, right? So now you have the probability of unaffected and also you have the probability of a girl child. The next thing I told you is that you have to find out which rule you have to use here: product rule, sum rule, or binomial theorem. These are the three main rules that we have studied in genetics, the probability rules. And if you remember, I have given you the keywords in the Brahma series session on Mendelian genetics. I gave you the keywords, and even in the class, those who are attending the regular classes, they know I always give a keyword so that they can remember, they can recall the concept faster. So product rule, you know, the keyword is independent event. For sum rule, it is mutually exclusive. And for binomial theorem, it's an unordered event. Now, you know, in the question, it is not an unordered event because it is specifically mentioned that first child will be an unaffected girl. So binomial theorem, we are not going to use. Also, it is not the case of sum rule because it's not a mutually exclusive event. Here, the independent events are given. Like if you talk about probability of the disease and probability of girl child, these are two different things, right? So independent events are there, and because they are independent events, we will be using the product rule. So probability of an affected child multiplied by probability of the girl child will give you 3/8, and this is the reason the answer here will be option 3. So Saujanya, I hope it is clear why we will not use binomial theorem here. And Surendra, yes, you are right. It will be a product rule. So majority of you have answered option 3. So absolutely correct. And Aishwarya, I hope now it is clear why the answer here is option 3. So moving over to another question now, which is again based on probability. Genes A, B, and C control 3 phenotypes which assort independently. Now, a plant with the genotype. So you can see this is the genotype of the plant and they are self. The question is, what is the probability? What is the probability for progeny which shows the dominant phenotype for at least one of the phenotypes controlled by genes A, B, and C? The options are given and you have to find out what is the probability for progeny to show the dominant phenotype for at least one of the phenotype controlled by genes A, B, and C. So how you are going to solve it? Believe me, these questions, they appear to be very difficult, but if your concepts are clear, if you know the strategy of solving the question, you can easily solve such type of questions. So in the beginning, well, when you are start practicing such questions, you can note down the information separately which is given to you in the question. So it is mentioned that there are 3 genes, A, B, and C, responsible for which control 3 phenotypes, right? And they are sort independently. So genes are not linked. And therefore, gene A is responsible for one trait, B for another, and C for another trait. Now, there is a plant which is triple heterozygous, right? And it is self. So, self, what kind of self? Like self cross will look like what? Like this, right? Now, you have one option, that is either you perform the, like you write down the all the genotypes possible in trihybrid cross, or the other way is you can use the tricks here. So what tricks you can use, I will tell you later, but like before that I want you all to solve it. And the third thing is that we have to find out that the probability of progeny showing the dominant phenotype for at least one of the phenotype. It means either See, you know that dominant phenotype it appears when we have the dominant allele in homozygous or heterozygous condition, right? So that means either we have the dominant allele for all the three genes either in homozygous or in heterozygous condition. Then only we'll get a dominant trait. Now combination can be anything. Like for A, we have a dominant allele, but maybe for B, we don't have a dominant allele, right? So the question is that at least the phenotype for at least one of the like the probability for progeny which shows the dominant phenotype for at least one of the phenotype controlled by A, B, and C. So either you can have this A in homozygous or heterozygous condition, and B could be in homozygous recessive and C also. Similarly, your A could be in homozygous recessive, but B should be like homozygous dominant and C should be homozygous recessive. So again, you'll get a dominant trait for B gene, right? Here you'll get a dominant trait for A gene. And same goes for C. So we can have different combinations, right? Because it's a trihybrid cross. So what do you think? What is the answer over here? Okay, so I can see you all have answered. Already some of you have answered. So it is option 3 you are answering. So let's check. So as I told you, it's a trihybrid cross and it is about the selfing, right? So if you remember, I told you one trick that either you can consider one gene pair at a time, right? So like if you consider this cross, it's a monohybrid cross, right? And this is another cross and this is another cross, right? So basically, other than instead of doing all these things, what you can do is, you know that if it's a trihybrid cross, how many gametes will be formed? So you know that we have a very simple mathematical rule here that we can use for determining number of gametes, that is 2 raised to power n, where n is number of heterozygous gene pairs, right? And you can see that here we have 3 heterozygous gene pairs, right? So here n will be 3. So n here is 3. So 2 raised to power 3 gives you what? 8. So that means 2 into 2 into 2 gives you 8. So 8 gametes are possible, right? And because we are performing selfing, right? So number of progeny also you can find out. So number of progeny in a trihybrid cross. So if you have studied trihybrid cross, you know it is 64. But you know the formula to calculate the number of progeny or offspring 4 raised to power n. That also you can use. So that will give you the number of offsprings. Now, in the progeny, I'm sure you know that one will be triple homozygous dominant and one will be triple homozygous recessive, right? And then you have different combinations. So that means only this combination is where you will never get a dominant phenotype, where all the recessive allele for 3 genes are present in homozygous condition. Here you will never get a dominant trait. So out of 64, in this one condition, you will not get, right? So that means out of 64, 63 you can get, right? And this is the reason we can say that because this is only one condition where you will not get a dominant for at least one gene. For like the question is, we should get at least dominant trait for at least one of the phenotype controlled by gene A, B, and C. So only in this condition we will not get a dominant phenotype. So here answer will be 63 by 64. So here you need not to draw the entire trihybrid cross or like individual monohybrid cross you are taking and you're finding the probability. No. So this is what I have explained you in the previous slide that when the parents, each parent will produce 8 gametes and total 64 progenies will be there. So one will be homozygous dominant, so triple homozygous dominant, and one will be triple homozygous recessive. And probability for at least one, it will be, that is rest of the individual other than this will give you, that is 63 will give you, will have at least one dominant character either in homozygous or heterozygous condition. So yes, you have answered it correctly. Saujanya and knew something. I hope it is clear why the answer here is option 3. Now moving over to another question. In the exam, you get questions on some hypothetical pathways as well, right? So here on your screen is one hypothetical pathway. And based on this pathway, there are some conditions given to us that there is allele, one allele converts X substance to H substance, another cannot able to convert it. So please go through it and you have to tell that which one of the following crosses can lead to above observation. So read the question carefully, see what information is given to you in the question, and then solve accordingly. So even in this pathway, you can see that X substance is converted by H allele to H substance. If instead of let's say this is a dominant allele, if we have this allele, so let's say, as you know, we represent the recessive allele with the lowercase letter. So the letter in lowercase form. So basically, this is a recessive allele, and it will not convert X to H substance, but it results into O phenotype. And then there is another allele A which converts H substance to A phenotype, and the recessive allele of the gene cannot convert it. Okay, if instead of this you have this allele, it will not convert it. Then if you have, so basically, if let's say instead of this you have this allele, so the pathway will be blocked. If the B allele converts H substance, so it converts it to B phenotype. But instead of this dominant allele, let's say you have a recessive allele, it cannot convert the H substance to B phenotype. Now, an individual with A phenotype is crossed with that of B phenotype, and they lead to a progeny that is with O phenotype. So, which one of the following crosses can lead to above observation? Okay, Rahul and Suresh, Achal, Surendra, Anupam, you have already answered option 2. Okay, so I'll give you some more time. I'll give some more time to the students the participants who have not yet answered the question. So some more time you can take, and then you have to answer it. See, answer is in the question itself. If you just follow the information given in the question, if you understand this hypothetical pathway thoroughly, you can easily answer this question. Okay, so I'm getting a uniform answer so far. That is option 2. Aruna, Anu, Aishwarya, you are also saying option 2. So that's great. So everyone has solved. Okay, so let me show you the answer and let me give you the explanation for the same. So basically, this is what is mentioned in the question, that we have a dominant allele which converts X substance to H. So here you have to consider that if a dominant allele is given, so that means either it is in homozygous condition or heterozygous. This thing is not mentioned to us in the question, right? So if the dominant allele convert X substance to H, that means either it will be in homozygous or it will be in heterozygous condition. But there is a recessive allele which cannot convert X to H, but convert it to O. And you know, recessive allele always express in homozygous recessive condition, right? The recessive allele, basically the phenotype of the recessive allele, we get in only when recessive allele is in homozygous condition. So here X is converted to O with the help of this allele. Now gene A convert H to A phenotype. So again, we don't know whether the dominant allele is in homozygous or heterozygous. So either this genotype could be there or this one could be there. And recessive allele cannot convert. So definitely it will be in homozygous recessive. Homozygous condition will be there. Then for gene B again, as it converts H to B phenotype, it will be in homozygous or heterozygous. And a recessive allele cannot convert H substance to B, so therefore it will be in homozygous recessive condition. Now, in the question, it is mentioned that A phenotype individual, when it is crossed with B phenotype individual, you get a O phenotype. Now, one thing is very much clear, that O phenotype you can only get if this allele is present, right? And this allele in homozygous condition should be there in the progeny. So that means the parents either must be like definitely they must be in heterozygous condition for this allele, or like H allele in heterozygous or homozygous recessive, right? So O phenotype is only possible when gene H is present in homozygous recessive condition, right? So in the progeny, there should be H allele in homozygous condition. And this is only possible when both the parents are contributing recessive allele at least, one recessive at least. So if the parents, just assume if parents are heterozygous, definitely each parent will contribute the recessive allele, and then only you will have a homozygous recessive condition. Because if you're considering one parent to be homozygous dominant, another homozygous recessive, you cannot get homozygous recessive in progeny, right? So they have to be in homozygous, or like any one parent can be in homozygous recessive for H allele. It can be possible. So we have to answer the question based on the options given to us. So definitely the genotype must be heterozygous. And if you look here in the options, one parent is having homozygous recessive, but another is homozygous dominant. So such combination will never give you the, like this combination will never give you the homozygous recessive. It will give you the heterozygous condition in the progeny, not homozygous recessive. Then yes, second could be the answer. If you see the third one, again, this one which I told you again cannot be the answer because another parent is not contributing recessively. And this one also cannot be the answer. So it's very simple. The answer here is option 2. Okay. So basically, you need not to be confused between A phenotype, B phenotype. Simply, if you focus on the H allele, you will simply get the answer in less time. So here answer is option 2. So everybody has answered it correctly. So that's great. Now, before moving ahead, I would like to tell you about Watson Junior Junoon Scholarship. And basically, the admissions are open for this Junoon Scholarship. So if you want to upgrade your current scholarship, and those who are not having any scholarship, they can apply for this scholarship. And the benefit of enrolling in this or taking this scholarship is that you get 1 year coaching for the national level examinations like CSIR NET, GATE, QET, IIT JAM, and GAT-B. So if anybody is preparing for IIT JAM, GAT-B, QET, other than CSIR NET and GATE, you can also inform it to your friends or your college mates. And also you'll get access, 12 months of access to global and national workshops, which are conducted by Biotechnica. You will be getting an R programming internship with live project hands-on, 4 certification courses of your choice. So through this scholarship, you can enroll to 4 certification courses. It completely depend on your choice, which 4 certification courses you select. And also you will get access to our experts on WhatsApp. And also you'll get a pass, VIP pass to the CRISPR conference which will be conducted most probably in November, CRISPR Congress 2023. And for more details, these are the toll-free number, and you can call on these toll-free numbers for more information. So let's move over to another question. In a certain breed of plants, thorns are determined by the dominant allele T, and thornless is determined by recessive allele, this one. Now, the T is 80% penetrant in heterozygote, and 20% of heterozygotes will appear thornless. In the monohybrid cross, what would be the expected ratio of phenotypes observed in a population of 500 F2 plants? Options are given to you. So you need to calculate that how many will be thorny and how many will be thornless. And this question is based on penetrance. So the concept of penetrance should be clear to you all. The penetrance, it is given in subunit C, that is extensions of Mendelian principles in your syllabus. So in the class, I have covered this topic in detail. So I hope you remember what is penetrance. Expressivity also I told you. So answer this question now. Okay, option one. So very quick, knew something. Okay, Uttara, you are saying option two. So yes, this question requires conceptual clarity and definitely you need to be thorough with what is penetrance. How will you calculate 80% of penetrance in heterozygote, 20% of heterozygote, right? That you have to carefully think about. Okay, Surendra, Anupam, Rahul, Aruna, you are mentioning option two. Fine. So I'm glad to see the answers. So far, uniform answers I'm getting. But whether this answer is correct or incorrect, that I will be telling you in some time. Okay, Anusha, Suresh, you are also saying option two. So what is penetrance basically? So let me first tell you about what is penetrance. Meanwhile, those who are still solving, they can write the answers in the chat. Okay. So penetrance is actually measured in percentage. And it is the percentage of individuals with a given genotype who exhibit the phenotype associated with that genotype. That means it's the percentage of individual that shows a phenotype which they are capable of showing. In a very simple sense, penetrance it is, we measured in a percentage, and the it we always denoted it in percentage, and we say that percentage of individuals they show the phenotype which they are capable of showing, then we say penetrance is 100%. Like for example, let's say there is the allele, let's say we have a allele, let's say B, for brown color of the leaves. And let's say this dominant allele is for brown leaf, and we have, let's say, the recessive allele for the white color of the leaf. Now, basically, let's say these all leaves that you see here are heterozygous. Okay. What is their genotype? They all are heterozygous. So if all the leaves here, they show the phenotype which they are capable of showing. So what they are capable of showing? They are capable of showing brown color, brown leaf color, right? So if they show the brown leaf color, that means we say that penetrance is 100%. Penetrance is complete, right? So like if any homozygous recessive, if any individual is having homozygous recessive condition and they show they are for a particular genotype, for a particular phenotype, and the individuals with this genotype show that phenotype, penetrance is 100%. Right? So when all homozygous recessive show one phenotype, when all homozygous dominant show another phenotype, right? Like obviously dominant trait, recessive trait will be there. Heterozygous are all same. Then we say that it's a 100% penetrance, complete penetrance is there. However, if less than 100% of individuals will are showing the phenotype which they are capable of showing, we say penetrance is incomplete. Like here you can see that all the, again, here all the leaves that you see here, they are all having the heterozygous condition, genotype. Okay, that is, they have the genotype heterozygous condition. So they all should show brown color of the leaf. But you can see that out of these leaves, two are not showing it. So in that case, penetrance is not complete, it is incomplete. Like ideally, they should show brown, but they are showing white, right? So therefore, penetrance is not complete. Now, if you ask me, ma'am, what is the reason for that? So remember, in Mendel experiments, we have studied that usually the gene is responsible for a particular trait, right? Like if anybody is having, let's say, a flower, a plant with a having a genotype for red flower color, so plant will bear the flowers which are red in color. But later on, several experiments when were conducted, it was observed that even environment has a role to play, right? So there are several environmental factors which are affecting the phenotype. Okay, so this is the reason that we are not observing here the trait which we expect that individual to show having a particular genotype for that trait. So it is mentioned in the question that when we'll perform the selfing of F1 generation. So you know that this is the parent generation. One would have been always homozygous dominant, another would be homozygous recessive. F1 generation will be heterozygous, and on selfing of F1 generation, we get the F2 generation, second filial generation, and the phenotypic ratio is always 3:1, right? So you know that in the case, like it's a monohybrid cross, right? So we know in a monohybrid cross, we get out of four, one progeny will show homozygous dominant, another will be heterozygous, another two will be heterozygous, and one out of four will be homozygous recessive, right? So here I have told you the probability of getting a dominant trait, recessive trait, and heterozygous condition, right? Homozygous dominant trait, homozygous recessive condition, and heterozygous condition. So phenotypic ratio is 3:1, but you can see the genotypic ratio is 1 out of 4 homozygous dominant and 2 out of 4 will be heterozygous. And if I further simplify, it is half. That is out of 4, half are heterozygous and 1 out of 4 will be homozygous recessive. Now, let's follow the question. In the question, it is mentioned that this particular allele is for thorns and it is 80% penetrant in heterozygote. So when you are performing a monohybrid cross, which I showed you in the previous slide, the probability of getting a heterozygous condition is what? Half. And the total offspring, the total progeny is 500 given to us in the question, right? So in a population of 500 F2 plants, how many will be heterozygous? So half of 500, that is 250. Why we are doing like this? Because in the monohybrid cross, heterozygous progeny is only half, 2 out of 4, right? So 500 F2 plants are there. So half of 500, that is 250, will be heterozygous. Now, in this heterozygous, only 80%, like this T is 80% penetrant. So 80% of 250 is how much? 200, right? So thorny phenotype observed in population will be, so because here dominant allele is present, right? So dominant allele, that is for thorns, you will get when you have the dominant allele in homozygous or in heterozygous condition, right? So we have 200, right? 200 in which this phenotype, like this T for thorns, which is 80% penetrant in heterozygote. So we have calculated 200, right? So 200 this and 125 for homozygous dominant condition. So 1 by 4th of 500 is how much? 125. So that means this much population is having homozygous dominant condition. So that totals gives you 325. Now, when I say 325, so obviously any option having the 325 for thorny will be the answer. But let's check for the recessive phenotype also. Recessive phenotype, so that means recessive phenotype, you know, it happens when you have a recessive allele in homozygous condition, right? So 1/4 of 500 is 125. So that means this much of the population is thornless. And it is also mentioned that 20% of heterozygotes will only appear thornless. So 20% of heterozygotes, again 250, right? So 20% of 250 is 50 now, right? So when you add 125 and 50, you get 175. So that is this much of the progeny will be, population will be thornless. And therefore, these options like 325 for thorny and 175 for thornless is given in option 2. And therefore, option 2 is the answer. So yes, you are absolutely right, everybody. Answer here will be option 2. So I'm really glad to see your responses. You have answered the question correctly. Now moving over to another question. In a flower species, light pink locus is hypostatic to pigment development locus. And this dominant allele, that is this R, is for pigment development, and the dominant allele W is for light pink. The recessive allele W in the light pink locus give red color, and recessive allele in pigment locus, it gives white color. So what will be the phenotype of this genotype which is given to you? So read the question carefully. It is based on epistasis, epistatic gene, hypostatic gene. You need to know here, and accordingly, you need to answer this question. So recall what is epistasis here and then solve the question. Yes, quickly answer the question. In one local, like this is the genotype given to you. So what will be the phenotype of this genotype? Okay, Achal, you have answered first. So option 4. Fine. What about others? Okay, Rahul, you are also saying option 4. Okay, again, uniform answer I'm getting so far. Knew something, Aruna, Anusha, you are saying option 4. Uttara, you are also saying option 4. That's great. Let's see if it is correct or not. So basically, what is epistasis? Epistasis, it is the interaction between alleles of two or more genes which are affecting the same character or control a single phenotype. So basically, in such kind of interaction between the, like we are talking about non-allelic condition here, where we are considering alleles of the different genes, two or more genes, and maybe these genes are responsible or they are controlling a single phenotype. So let's say we have two genes, and I'm writing the alleles of the first gene, which is this one, and the allele of another gene is this one. So when a allele of one gene masks the expression of another gene, either of the another gene, alleles of another gene, we call it as epistatic gene. However, a gene whose expression is masked, we call it as hypostatic. Okay, like for example, if A allele, this dominant A allele is masking the expression of either of the allele of another of the gene at another locus, then A allele is epistatic and B is hypostatic. Now we have different type of epistasis: dominant epistasis, recessive epistasis, duplicate dominant, duplicate recessive epistasis, and many more. So particularly if we look at this question, if we see this question, it is mentioned that R is for pigment development. So as I always say that write down the thing separately, the information separately, which is given to you in the question. This will help you understand the question thoroughly. You will not get confused. So R is for pigment development and W is for light pink. Light pink is hypostatic to pigment development locus, that is R, right? Now, there is a recessive allele in the light pink locus which gives red color, and recessive allele is there in the pigment locus which gives white color, right? So as you know that the pigment development locus is epistatic, so definitely here you will never see a color, right? You will always see a, like, you will never see a red color or pink color, you will always see a white color, right? Because at the pigment development locus, because it is epistatic, it will be epistatic to the dominant or recessive allele present at the light pink locus, and this is the reason here the flower color will be white, and the answer here is option 4. So you all have answered correctly, very good. Yes, Anupam, it will be option 4. Moving over to another question now. Following is a hypothetical biochemical pathway responsible for pigmentation of leaves. The pathway is controlled by two independently assorting genes A and B encoding enzymes as shown below. And so this is the pathway which is shown here. And mutant alleles code for non-functional proteins. So here you can see a substrate X is white in white, and it is converted to an intermediate in this biochemical pathway Y, which is yellow. And further, this yellow is converted to final product, which is green. Now, the A allele, okay, the dominant A allele is responsible, the normal allele we can say, because here mutant allele is mentioned. So, the normal allele A is responsible for conversion of the substrate into an intermediate, right? The precursor into an intermediate. And finally, intermediate will be converted by the product of gene B. Now, it is mentioned that if the mutant allele A and B, they are present, they code for non-functional protein. So basically, let's say this A gets mutated. So instead of the dominant A, if you have the mutant A, the mutant gene A will not able to encode for its functional product and pathway will be blocked here, even if you have a normal B allele. Because the product of gene B can only act on an intermediate. But if your A gene is mutated, the pathway itself will be blocked here. No intermediate will be formed. Similarly, if you have the normal A, but your B gene gets mutated, normal allele A, but B gene gets mutated, your substrate will be converted to intermediate, but further the final product will not be formed because the gene B here is mutated, and because of that, it will not encode the functional product, which is converting the intermediate into final product. So this is the explanation of the question. Please go through it and tell me what is the expected progeny after selfing a plant with genotype given. So this is the double heterozygous condition mentioned, and you can see this kind of selfing has to be need to be done, and you have to find out what ratio you will get. So usually if you see in Mendel's dihybrid cross, we always get phenotypic ratio 9 is to 3 is to 3 is to 1. Mendel's dihybrid cross always give 9 is to 3 is to 3 is to 1 ratio. But here it is the case of epistasis, right? Like if you have studied epistasis in detail, you will come across a lot of these biochemical pathways, and different type of epistasis are there. As I told you earlier, we have duplicate dominant epistasis, recessive epistasis, dominant epistasis, duplicate recessive epistasis, polygenic gene interaction, many are there. So if you have studied those epistasis in detail, you can easily answer this question. And in the class, I have covered this topic in detail. So here I'm getting the option 1 as a uniform answer. I'm getting Suresh, Surendra, Achal, you all are answering option 1. Okay, so let's others also solve and I'll then tell you the answer. Okay, Rahul, you are also saying option 1. So pathway you all understood, right? So one thing is very much clear that if you have the dominant A and B allele present in the progeny, then only you will have a green phenotype, right? You will get a final product Z, which is green, right? So for that, we need to have dominant allele, either in homozygous or heterozygous condition, both in homozygous or heterozygous, or in one in homozygous, another in heterozygous, right? Another one in homozygous, another in heterozygous, or vice versa, like that. Okay, so everyone is answering option one. Let's check. So let me tell you, this is an example of recessive epistasis. So as I explained you in the previous slide, that the white precursor is being converted by the normal allele, dominant allele A. And if this allele gets mutated, definitely the pathway will be blocked here, right? And this yellow, the intermediate which is formed, is converted into the final product, which is green, by the gene, the product of gene B. But if this gene B gets mutated, you have a mutant allele, definitely pathway will be blocked here, right? So here we have to perform the selfing of this individual with this genotype. So you know that it's a dihybrid cross, and you always get 16 offsprings in the F2 generation, right? So how many of them will be green? How many of them will be yellow or white? That we have to find out. So obviously, as I told you, if the dominant A and B alleles are there, either both are in homozygous, or like both are in heterozygous, or any one in homozygous, another in heterozygous, or vice versa, then we will get only the green pigment. If you have the B allele mutated, so A in homozygous or heterozygous, but B is mutated, the pathway will be blocked here. So in that case, you'll get yellow pigment. If this is the genotype, you'll get yellow pigment, which is shown here. So wherever you have homozygous, the B allele, recessive allele in homozygous condition, you will get yellow pigment. But if you have the A allele in homozygous condition, right? So this allele is recessive allele, right? And it is mutated, so pathway will be blocked here. It doesn't matter even if you have a dominant normal B allele. So here you have a normal B allele in homozygous or heterozygous, but because the mutant A allele is there, pathway will be blocked here. So we can say the recessive A allele is epistatic to the dominant or the dominant allele of the another gene, or recessive allele of the another gene. So this results into 9 is to 4 is to 3 ratio, that is green 9, white 4, and yellow 3. So usually, you know, if a dominant allele is there, it should show its expression, right? So we have a dominant B allele here. So we should get yellow. But we are not getting yellow here because of the epistasis. We can say that this recessive allele A is epistatic, or it masks the expression of the dominant and recessive allele of the another locus, that is the dominant B and the recessive B allele. It's an example of recessive epistasis. And now let's move over to another question. So answer here is option 1. Uttara, I hope it is clear why answer here is option 1. So let's discuss the last question of today's session. So the question is that in rabbits, one enzyme, the product of functional gene A, is needed to produce a substance required for hearing. Another enzyme, which is a product of gene B. Okay, so there is a gene A and its product is required to produce a substance for hearing. Okay. Similarly, there is another enzyme which is encoded by the gene B, and it is also needed to produce another substance required for hearing. So we can say that it's a pathway, maybe a pathway where we have a precursor which is converted into the intermediate by the product of gene A, and this intermediate is finally converted into final product by the product of gene B. And this product which is formed is finally been responsible for hearing. So that means we need both gene A and B for hearing. So that means if any one gets mutated, there will be a problem. So genes responsible for two enzymes, they are not linked, right? So they are independently assorting. And individuals homozygous for either one or both of non-functional alleles A or B are deaf. So that means if you have this condition, if this condition is there, homozygous recessive A or B, right? So homozygous for either one or both. That is, either here we have this condition, or like any one homozygous, or here like anyone, or both. So in such condition, the pathway will not be completed and definitely we'll get the individual which is deaf. Now, when a large number of matings were made between two double heterozygotes, 9 hearing rabbits and 7 deaf rabbits is observed in the progeny. And the phenotypic ratio found is 9:7. So what this 9:7 ratio is, so what is this phenomenon? What is this well-known phenomenon? So you can see in the previous question, you need to find out the ratio. Now, in the exam, CSIR NET exam, instead of giving the question like this, now they have given you the ratio and they have asked you what kind of epistasis it is, right? So, question can be asked both ways. Either they'll give you like some biochemical pathway and they'll give you the, like you from that biochemical pathway, you have to recall what kind of epistasis it is, and then you have to choose the ratio in the options given. However, the another way of doing it is like what they can do is they can give you the entire experiment and they'll give you the ratio and they'll ask you what kind of epistasis it is. So please answer this question. Okay. So option 4, duplicate recessive epistasis, everybody is saying. Suresh, Anu, Achal, Rahul, Anusha, Aruna, Anupam. I must say, students, dear participants, I really appreciate you all for answering, taking out time and attending the sessions. And I'm really glad that you are taking the initiative and trying the questions. And I'm sure, I need not to tell you, you are answering it correctly. So I'm sure this will definitely give you a confidence, right? So I can say that this Brahma series is definitely helping you all. Are you all agree with me? Are you enjoying these Brahma series sessions? Are you finding them helpful? Please let me know in the chat. So definitely, I must say today, whatever answers you, most of you have answered the questions correctly. So very good, keep it up. I'm really proud of you all. Okay, thank you all for confirming. Thank you, Achal. Thank you, Rahul, Anusha, knew something, Aruna, Surendra, for your response. So yes, you all have answered option 4. Yes, Anusha, so you are able to revise also. That's great. Good to know. Yes, Anupam, good for revision. Definitely. Okay, thank you, Uttara, for your response. You find it helpful, so really good to know about it. So yes, from the ratio, coming back to the question, so from ratio 9:7, we can directly say what kind of epistasis it is, right? So as most of you are saying that it's very good for revision. So here is one table for quick revision, right? So 9:7, it is about duplicate recessive epistasis, right? Where like the recessive allele of one gene is epistatic to the either of the allele of another gene, or the recessive allele of another gene is epistatic to either of the allele of another gene. So duplicate recessive epistasis it is. So directly from the ratio, you can answer the question. And if you want to understand the question in detail, so basically, as I mentioned earlier, that we need both A, dominant A and B allele. So if they are in homozygous or heterozygous condition, or any one is in homozygous condition, another in heterozygous or vice versa, we'll get the individual will be here, will not be deaf, right? But anywhere you are getting the recessive allele, either B allele in homozygous recessive condition or A in homozygous recessive, that leads to the deafness. So individuals are deaf if they are homozygous for either one or both recessive allele, right? So it's an example of duplicate recessive epistasis, which I told you that recessive allele of one gene is epistatic to the dominant allele or either allele of another gene or vice versa. So here the ratio is 9:7. 9 will be hearing and 7 will be deaf. So answer here is option 4. So you all have answered it correctly. So very good. Keep it up. So with this, I have come to an end of this session. And one suggestion I would like to give you all in the end, that success is a state of mind. So if you want success in CSIR NET exam, start thinking of yourself that you have qualified CSIR NET exam with a good rank. And once you start thinking like this, definitely you will achieve success. So this is all for now. Thank you, everyone. And we'll see you in another session. Take care.